Hello class, it's Mr. Mormon. In this video we will be reviewing for your midterm. The midterm this year is going to be a 2-day in-class event. Let's start with one of the first things we did this year. You need to know the difference between observation and inference. When we make observations, we're using our senses to gather information: seeing, hearing, smelling, tasting, touching. And inference is when we go beyond our observations. It's when we interpret or explain our observations. Okay, let's recall some of the things that we learned about the metric measurement system. When you're measuring length, you're measuring the longest dimension of an object. Mass is the amount of matter in an object, volume is the amount of space it takes up, and density you don't measure directly. Density you actually calculate. It's the mass divided by the volume. What you're really figuring out is how well packed the matter is, how much atoms and molecules are packed in every given bit of space. You also need to be familiar with the tools and units for each of these things that we measured. Length can be measured with ruler, tape measure, or odometer. The unit for length is the meter. We can use smaller units like centimeter and millimeter, 1/100th of a meter, 1/1000th of a meter, or larger units like kilometer, which is 1,000 meters. Mass is measured with a triple beam balance in grams. Milligrams would be 1,000th of a gram. Kilograms would be 1,000 grams. Volume of a liquid can be measured with a graduated cylinder, or we can do length times width times height. There's formulas for different regular shapes. This is the formula for a rectangular prism or a cube. The meter for liquid, the unit for liquid is liters. A milliliter would be 1,000th of a liter, and a cubic centimeter is actually the same volume as a milliliter. Density is mass divided by the volume. Grams per cubic centimeter would be 1 unit, or grams per milliliter. Okay, let's practice real quick. Let's, why don't you pause the video and give me an answer for A through H. What would your measurement be? Notice that each of these measurements I estimated to the nearest tenth of a centimeter, and whenever you write a measurement, you always include the units. Also on this one, notice it came exactly to 9. This one that came exactly to 9 centimeters, I wrote as 9.0. Always record your answer to the tenths place. It shows the accuracy that you measured to. Okay, mass is measured with a triple beam balance. You put the object on the pan and then move the riders, starting with the largest one, to balance it out. You then read the riders, add them up, the weights that are on the beams. You add them up for the total mass. So what would the mass of this object be? Again, we measure to the nearest tenth of a unit. These units would be grams, of course, and we add this up, 175.0 grams. Now, what would your answer be for these practice problems? This first one, 288.2. This one looks like 165.9. Again, make sure you're including the units. This last one, 511.0 grams. Now, when measuring the volume of liquids, you're going to use a graduated cylinder. Just put the liquid into the cylinder. Because of cohesion, the liquid will be pulled up the sides a little bit, making this kind of U shape, which we call a meniscus. Remember to read the bottom of the meniscus. Regularly shaped solids usually have a formula. Like here's the formula for a rectangular prism. It's length times width times height. Your units for the graduated cylinder will be milliliters. The units for a regularly shaped solid will be cubic centimeters. Remember that they're actually the same volume. A milliliter equals a cubic centimeter. Now, let's talk a little bit about density. With density, you're measuring how well packed the matter is. It's calculated by the mass divided by the volume. And here's a good example that kind of displays what density means. These two objects are the same mass. They're both 20 grams, but we can see A is much bigger. It must be less dense. The material is not as well packed as it is in B, where there's more matter in a smaller space. B would be more dense than A. Their mass is equal. B's volume is much smaller. Another thing that you should remember about density is things that are less dense than water will float, things that are more dense than water will sink. Okay, when doing a good controlled experiment, we're going to use the scientific method. A good controlled experiment tests only one thing at a time, one independent or manipulated variable. We're going to keep all other variables constant. And the thing that you measure in the experiment is your dependent or responding variable. Now, in the scientific method, we start with a problem or question. We then do research and come up with a hypothesis. This is what we think the answer to the problem is. We'll often write it as an if-then statement. You then are going to design your experiment where we will test only one factor. And that one factor is your manipulated or independent variable. The respondent or dependent variable is what you measure in the experiment. You then perform the experiment, collect your data. Often it will go into a data table or be placed on a graph. And from that you would form your conclusion, where you answer the question, does my data support my hypothesis? A good controlled experiment will also often have a control group. This is where you do part of the experiment, but you leave the independent variable out. This is your normal setup that you are not really testing. You're running this one as a comparison to see if your independent variable is doing anything at all. All right, let's review some stuff about graphing. First of all, the axes. This is the y-axis. This is the x-axis. You then have to determine what variable you're going to place on what axis. The simple phrase dry mix can help you remember this. The D and the R stand for dependent and responding. The M and the I stand for manipulated and independent. Remember to always include a title with your graph. You need to label the variable in units on each axis and then create a scale. You're going to use a consistent scale on each axis. They do not have to be the same. You'll usually start with 0, and here we're going by 1s. You can also go by 2s, or 5s, or 10s, or whatever interval best fits your data. Again, the scale needs to be consistent. It's got to be, you can't start going by 2s and then realize you're running out of room and switch to going by 5s. It's got to be the same scale the whole length of the axis. Again, each axis could have a different scale. That's okay, but the scale on the axis needs to be consistent. Don't forget to label your units, the variables and the units on the axis. Okay, let's practice plotting data. Here we have a data table. This is water temperature, and this is how much solute will dissolve at that temperature. We need to graph it on this graph paper. The first ordered pair is 0 degrees Celsius and 30 grams. So I find 0 degrees Celsius down on the bottom, go up to 30, and there's my first data point. At 20 degrees, it's 37. So I find 20 degrees. I follow that line up. This would be 30. 35, 37 would be about there. At 40, it's 46, maybe somewhere around there. Now read the directions carefully. Often it says to circle your data points, and then you finish up by connecting them with a line. Now that graph was about solubility. Solubility is a measure of the amount of solute that can be dissolved in a solvent. It can be increased by heating, crushing the solute, and stirring the solvent. Each one of these things will increase solubility. So in this example, which one do you think would have the highest solubility? I choose this one. It has both a crushed solute and a higher temperature. If I stir it, it will dissolve even faster. Okay, quick review of atoms and molecules. Atoms are the tiny particles that everything in the universe is made of. Remember that they contain different parts. There's a nucleus in the middle with protons and neutrons in it, and electrons are whizzing around the outside. Protons and neutrons have mass. The electrons have almost no mass. Remember that the protons have a positive charge, the electrons have a negative charge. Neutrons have mass, but they have almost no charge. They have the same mass as a proton, 1 AMU, atomic mass unit. Electrons are about 2,000 times smaller or less massive. Now, atoms join together with chemical bonds to form molecules, like we see here in these models. Okay, do you remember the difference between a physical and a chemical change? In a physical change, you just change the shape or form of the material, but it still remains the same material. In a chemical change, chemical bonds are broken or new ones are formed, and you end up with a new substance. Examples of physical changes would be crushing, tearing, ripping, chopping wood. Phase changes like freezing, melting, those are physical as well. Burning, rusting, cooking, each of these, digesting food, each of these ends up with a new material. So those are chemical changes. Okay, remember that matter is not created or destroyed in a chemical reaction. You always have the same amount of matter, or you can measure it with mass, before and after a chemical reaction. Now, sometimes it doesn't seem like that, like when you think of maybe a log burning up in a fire. It seems like the ashes have much less mass than the log you started with. And that's true, but don't forget about all the carbon dioxide and all of the other things that escaped into the atmosphere during the burning. None of those atoms were destroyed. They're all still, in the atmosphere. So the actual mass or matter is the same as when you began. Okay, let's review some things that we should remember about life. These are characteristics and needs of living things. First of all living things are made of cells. Some are multicellular, some are unicellular. Unicellular means they only have one cell. Cells come from other cells. Life comes from life. There's no such thing as spontaneous generation where life springs up from non-living matter. Life always comes from life. Living things use DNA as a blueprint, a set of plans, or genetic instructions on how that organism gets put together and how all of its parts function. Living things respond to stimuli in their external environment. Living things change over time. They evolve to become better adapted to their environment. Living things transport material in and out of their bodies or around through their cells. All living things need energy. Some get their energy right from the sun, others get their energy by eating. And all living things need water. Water is the main ingredient in most living things, 70% or more. Do you remember the life processes? We remember them with this silly little phrase, 10 GERS. Okay, there's lots of things that we need to remember about photosynthesis and cellular respiration. Photosynthesis is done by plants. We would call them autotrophs or producers because they are actually producing their own food and become the food source for everything else in an ecosystem. Cellular respiration is performed by both plants and animals, autotrophs and heterotrophs. The difference is where the food or the fuel to power respiration is coming from. Autotrophs are making their own, heterotrophs need to eat. If we were to define photosynthesis, it captures sunlight energy and stores it in the chemical bonds of glucose. Glucose is cell food. The purpose of cellular respiration is to release that energy stored in the chemical bonds of glucose. Again, the difference between plants and animals is where they get the glucose from. Plants make it themselves in photosynthesis. Animals need to eat. Okay, let's recall the cell structures where this is all happening. In plants or autotrophs, photosynthesis is happening in the chloroplast. Cellular respiration happens in the mighty mitochondria. Now, the raw materials a plant needs, carbon dioxide and water. Of course, they're going to need sunlight energy also, but that's not a material. What's being produced is glucose and glucose and oxygen are the products of photosynthesis. Photosynthesis is on the left here. Cellular respiration, the raw materials are glucose and oxygen. Glucose is the fuel. You need oxygen as well. After the glucose gets broken down, you're left with carbon dioxide and water. Here's a diagram showing the raw materials and products for photosynthesis. Of course, whenever you see a leaf in a problem, that should remind you that we're probably talking about photosynthesis. Got sunlight energy coming in, carbon dioxide out of the air, water up from the roots and through the stem. The sugar is produced and oxygen is released by the plant because they do not need that oxygen. Now, don't make the mistake. A lot of students say that the reason plants are doing photosynthesis is to make this oxygen. It's not. It's to make the sugar. Oxygen is a waste product. They don't need it. They don't want it. They get rid of it. That's good for all of us oxygen breathers, though. This is the diagram of for cellular respiration. We see the food and oxygen in. Of course, that food would be glucose. Energy is released from that glucose. Carbon dioxide and water are given off from the cell as waste. The water gets recycled. The carbon dioxide is exhaled or just goes into the environment. Okay, remember that unicellular organisms have organelles that perform all their life functions, where a multicellular organism has organs to perform all its life functions. Okay, recall that a microscope does several things to an image. It increases the magnification, making it look bigger, increases the resolution, making it look sharper so you can see more detail. It also reverses the image, making it upside down and backwards. Recall how to make a wet mount slide. The specimen goes in the middle of the slide. You add a drop of water. The cover slip gets gently placed down at a 45-degree angle to eliminate air bubbles. If you need to add stain to make the cells more visible, you would make the wet mount slide, add the stain to the edge of the cover slip, and then draw it underneath by placing a paper towel on the other side of the cover slip, and it will, by capillary action, pull the stain across. This will make your cells or parts of the cells more visible. Okay, you should be able to tell the difference between plant cells and animal cells. This picture is of a plant cell. I know that because of its boxy shape. That's one thing. It has a thick cell wall around the outside. Animal cells do not have that. I see this large water vacuole, which an animal cell would not have. Animal cells have vacuoles, but not a large water vacuole. I also see chloroplasts all the way around. If this was a color picture, these would be green. Now, things that would be the same between a plant and animal cell, well, there's a cell membrane, and that would be the outside layer of an animal cell. There's cytoplasm. There's a nucleus here in the middle. A lot of the other organelles, which we don't see here, would be the same as well. Ribosomes, endoplasmic reticulum, mitochondria, you'd find in both kinds of cells. Okay, you should remember the levels of organization. It starts with cells. It's the smallest unit of life. Cells are grouped together. The same kind of cells doing a job would be called a tissue. You can talk about muscle tissue, brain tissue, bone tissue. Even blood is considered a tissue. Tissues are grouped together to become organs. The heart is made of several different kinds of tissues, for example. The heart's got muscle tissue, connective tissue holding it together, nerves, there's blood in there, all different kinds of tissues working together to do a job. Organs are part of organ systems, and put all the organ systems together and you have the whole organism. All right, let's cover some ecology ideas. Remember that in an ecosystem, energy moves through an ecosystem and is used up. And we could show that with food chains and food webs. Remember that the arrows show the way the energy is flowing. Also remember that the higher up the food chain you go, there's fewer and fewer organisms. These high-level predators would be the most rare in the ecosystem. They would also contain the least energy, and we would show that with an energy pyramid. The producers down here at the bottom would be the most in number and also contain the most energy in the ecosystem. We could identify these animals as herbivores because they're eating plants. These up here would be carnivores because they're eating animals. An omnivore would eat both plants and animals. And don't forget about decomposers. These would be fungi and bacteria that are breaking down all the dead matter and recycling the nutrients in the ecosystem, a very important part of any food web. Recall that matter is a little bit different how that moves through an ecosystem. The energy gets used up, but matter, because it cannot be destroyed, it's just recycled. Plants are producing oxygen that are then used by animals. Animals produce carbon dioxide, which are used by the plants. And these atoms and molecules just exchange places and move around the ecosystem, but they get recycled. The water cycle is another example of that. The water can be found in a lot of different places as rain, up in the clouds, groundwater, and it cycles from one place to the other. Okay, we're almost done here. We could use this little ecosystem to review a few other ecology concepts. Again, sunlight coming in. The plant is the autotroph or producer, collecting that sunlight energy. There would also be algae in here, microscopic plants. They become, they're the producers. They're producing oxygen for the animals. They're also become food for the animals. The animals would be herbivores, like these snails could be eating that algae. They would be herbivores. And this fish is actually probably a carnivore, but you'd have to add food for the fish to eat. So the energy gets used up, the matter gets recycled. Oxygen released from the plants, carbon dioxide released from the animals, and it just moves around through the ecosystem. Okay, I think that's about it for this review. If you have any questions, you can ask me in class.